Ever wonder if there's a master blueprint for every single living thing on our planet? A single microscopic instruction manual that decides everything from the color of your eyes to how a giant redwood tree grows? Well, it turns out there is. And for a very long time, the biggest question in biology was just trying to figure out what this code is and how in the world it works.
So that was the big mystery that had scientists scratching their heads for decades. And in this explainer, we're going to unpack the incredible story of how they finally found the answer, how that answer actually works, and how it builds everything from a tiny microbe all the way up to you and me.
All right, so our journey starts with the blueprint itself. Turns out all of those instructions for all of life are packed into one unbelievable molecule. Okay, get ready for this, because the scale we're talking about is just mind-blowing. That number right there, 2.2 meters, that's the length of the DNA crammed inside just one of your cells, and you've got trillions of them. So how do you pack a 2-meter-long string of information into a space smaller than a speck of dust? I mean, it is an absolute marvel of natural engineering. And that incredible molecule is, of course, deoxyribonucleic acid. You know it is DNA. Just picture a super long chain, a polymer that's made up of smaller repeating units. This one chain is the ultimate genetic material for pretty much every living thing on Earth.
Okay, so knowing the molecule exists, that's step one, but understanding its shape. Now that was the key that unlocked everything else, and it kicked off one of the greatest scientific races in all of history.
You know, this slide really shows you that science isn't just one single eureka moment. It's more like a relay race, with the baton being passed down through generations. It started way back in 1869 with Miescher just finding some new stuff in a cell's nucleus. Then you had these huge experiments from people like Griffith, Avery, and Hershey and Chase, who proved, step by painstaking step, that DNA was the real deal. All of it leading up to that famous moment in 1953 with Watson and Crick's model.
So, to really get this structure, you got to break it down to its most basic piece, the nucleotide. It sounds complicated, but it's actually pretty simple. Every single link in that long DNA chain is made of just 3 parts, a sugar and a phosphate group, which form the backbone, and then a nitrogenous base. And that base, that's the part that carries the information. It's the actual letter in the genetic alphabet.
And those bases, they come in 4 flavors, if you will: adenine, guanine, cytosine, and thymine. But we can just call them A, G, C, and T. Think about that for a second. The entire complex instruction manual for building you is written with just these 4 letters. That's it.
Okay, but here's the real breakthrough, the secret sauce. It came from figuring out how these letters pair up, because it's not random at all. A scientist named Erwin Chargaff discovered a golden rule. A always hooks up with T, and G always hooks up with C. And this one simple rule, well, it is the key to absolutely everything.
So when you put all of that together, you can see how the final structure is built. Those AT and GC pairs, they form the rungs of a ladder. The sugar-phosphate backbones make up the sides, and then, as Watson and Crick showed the world, you give that ladder a gentle twist and voila, you get this, the iconic double helix. It's an elegant, stable, and frankly, beautiful architecture, just perfect for storing the blueprint of life.
A master plan is great, but it's kind of useless if you can't make copies, right? I mean, every single time one of your cells divides, it needs a complete, perfect copy of the entire instruction manual. So, how does life do it? Watson and Crick totally knew how huge this was. In their 1953 paper, they dropped this little gem. It's got to be one of the biggest understatements in science history. What they were saying in their very reserved way was that the very shape of the molecule was screaming the secret of how it copies itself. That base pairing rule was the clue.
The process has a bit of a mouthful of a name: semi-conservative replication. It sounds complex, I know, but the idea behind it is actually beautiful and super simple. Semi-conservative just means that in every new copy, half of the molecule is old, the original, or conserved half, and the other half is brand new.
So how does this magic trick actually happen? It's really just 3 basic steps. First, cellular machinery unwinds the double helix and unzips the 2 strands, just like a zipper. Second, each of those original strands now serves as a perfect template. And third, the cell's machinery just builds a new matching strand on top of each template, following that golden rule: A pairs with T, G pairs with C.
And just like that, from one DNA molecule, you get 2 perfect copies.
Okay, so we've got the blueprint. We know how to copy it. Awesome. But now for the big question. How does the cell actually read the darn thing and start building stuff? How does a long string of A's, T's, C's, and G's actually become a working part of you? Well, this whole process is guided by something Francis Crick famously called the central dogma of molecular biology. It's the fundamental flow of information in a cell. The permanent master blueprint, the DNA, is used to make a temporary message, which is called RNA. And then that RNA message is read by the cell to build a protein. And it's the proteins that do all the heavy lifting in our bodies.
Here's a great way to think about it. DNA is like the priceless, original architectural blueprint that you keep locked safely away in the main office. That's the cell's nucleus. You would never take that original out to the messy construction site, right? No way. Instead, you make a cheap, disposable photocopy to take with you. That's RNA. It's a temporary messenger. It's single-stranded, and it uses a slightly different letter, uracil or U, instead of T, to carry the message out of the nucleus to the construction site.
And this all goes down in 2 key stages. First up is transcription. That's where a specific gene on the DNA is copied into that messenger RNA or mRNA molecule. Then comes translation. This is where that mRNA message is carried over to a little cellular factory called a ribosome, and the ribosome reads the code and builds a protein, one piece at a time.
But wait, how does the ribosome actually know what to do? How does it read the message? It uses something called the genetic code. And here's the secret. The ribosome doesn't read one letter at a time. It reads in groups of 3. Each 3-letter word, called a codon, tells the ribosome to grab one specific amino acid. For example, the codon AUG always means start here, and the first amino acid is methionine.
Now, this whole system is unbelievably precise. But what happens when there's an error? A typo in the blueprint? Well, as you can imagine, the fallout can be massive.
Let me show you what I mean with a super simple analogy. Imagine our genetic code is just an English sentence made up of only 3-letter words, like the one you see here. Ram has red cap. The cell's ribosome reads this in a very specific frame, 3 letters at a time. Simple enough?
Okay, now watch what happens if we mess it up. Let's just stick a single letter B in there. Because the ribosome is programmed to always read in groups of 3, the entire reading frame gets shifted over by 1, and the message becomes total gibberish, Ram has breaket DCet P. Everything downstream from that one mistake is now completely scrambled. That's a frame shift mutation, and it can totally wreck the instructions for a protein.
And believe me, this isn't just a word game. A single typo in your DNA sequence, one wrong letter out of billions, can cause a devastating genetic disease like sickle cell anemia. It changes just one amino acid in a super important protein, but that's enough to warp the shape of red blood cells and cause all kinds of problems. It's a powerful example of why accuracy in this system is just so critical.
You know, for most of human history, this code was a complete mystery. We spent 100 years just proving DNA was the blueprint, and then decades more learning how to read it. But now, with game-changing tech like CRISPR, we're not just reading the code anymore, we're learning how to write it. And that leaves all of us with a really profound question for the future. What will we build?